plants have to have these microbes. They're microbial cultivators. They get their nutrients from microbes. They get the health from microbes. Their immune system is directly from those microbes. Their nutrients are from those microbes. And I consider that very important, supporting that group, because it's that group, the regenerative agricultures and the organic agricultures, that are going to actually change agriculture. So what support that I can give to them in this biological agriculture community, it can help them to be successful in changing agriculture for the sake of human health and the sake of environment and the world and better everything in general. Yeah.
You're not going to get rid of all the bugs, all the pests, and all the disease by treating your soil and treating your plants from the outside in with inputs and pesticides and herbicides. The way you're going to help your plants combat diseases and pests is through growing healthy plants, having biodiversity in your soil, and creating an environment where plants can flourish in the rhizophagy cycle, where they eat bacteria and don't absorb bacteria. We used to think that plants absorb bacteria, but it's just not true. They actually eat it, and they eat algae as well. In this conversation with Dr. James White, we explore all the different ways that plants are evolving in their immunity and in their fertility and in their health and even in their biodiversity in different climates. It's absolutely fascinating how plants actually cultivate the nutrients that they need. And it starts in the fertility of the soil, and it starts in the biodiversity of the soil. Dr. James White is a professor at Rutgers University. He's an extraordinary human being, and we are deeply honored to have him joining us for the Soil Summit 2024 Biodiversity, Cultivating Life. Enjoy the conversation.
Hi, Natalie.
Hi, Dr. James White. How are you?
I'm good. How are you?
Great, thanks. It's wonderful to be in the room with you again.
Oh, it's wonderful to see you again. In this virtual room,
In the virtual
Yeah,
Zoom
yeah.
room across the ethos.
We're working on this process called the rhizophagy cycle, like rhizo for root, phagy for eating, right? Where the plant roots actually absorb microbes from the soil and then extract nutrients from them, and then put microbes, that this is happening at root tips, right? Absorb them into the root cells and then eject them out of the root hairs, the tips of the root hairs. So they go back to the soil from the root hairs as those hairs grow. And they carry nutrients to the plant. So we work on that process, trying to figure out what's involved with it, what microbes are involved. We recently found that algae can actually be absorbed into roots and into the root cells. And when they do, they actually carry bacteria with them, because the algae have their own bacteria, their own endophytes, endophytic microbes. These are microbes that go into plants and plant cells. And so when the plants absorb algae, they degrade the algae, the algal cell, and then they get the bacteria that the alga had, and they take that bacteria and they use that for themselves to trigger their own growth and development, and to acquire nutrients for them in the soil. So by consuming algae, they have a way of not only getting nutrients, but getting other endophytes, other microbes that they could then use. It's really interesting to explore this stuff.
That's fascinating, because the last time we spoke, we were just talking about the plants eating bacteria, and I'll say eating bacteria. And now you've learned that they actually also eat algae. And
they can eat algae. Yeah.
Fascinating. And what's in the algae that the plants like?
Well, I mean, you could consider everything they're made of. I mean, considering the chlorophylls, you're going to have magnesium, right? Magnesium, and lots of other nutrients, lots of nitrogen in them, lots of phosphorus in them. So and lots of other micronutrients. They break these algae down and they get all of those nutrients from the algae. Not only that, because the algae are growing out in the soil with the plant, some of these algae are soil algae, right? And so because they're growing in the soil around the plant, it's a source of endophytes for the plant. So it's like a cover crop that has endophytes in it that grow around the crop plant, and those endophytes can go out from the cover crop and go into the crop plant and stimulate the growth of the crop plant, right? But in this case, the alga grows down there. It's just a tiny little cover crop, right? Right down there near the root. And so the plant can then absorb that and get all those, get the nutrients from it and get the bacterial endophytes out of it. So it is amazing, like a interesting little cover crop that grows right around where the plant roots are.
Yeah. And remind us what endophytes are.
Okay, endophyte are the microbes that go inside plant roots, cells, leaves. They go inside the plant, right? Inside the tissues and the cells of the plant. That's an endophyte. Internal, E-N-D-O, internal, phyte, P-H-Y-T-E, plant. Endophyte, plant, internal plant. Okay, so any microbe that goes in a plant is an endophyte. And it doesn't mean it's a special microbe. It just means it's in the plant took it in and the plant has it inside it. And it turns out that's how plants work.
And it's helping the plant. It's good for the plant. The plant's
They
own.
help. Yeah,
Yeah.
it's
Like
good
building blocks, building bricks.
Yeah, the plant has to have them. Yeah.
So two things come to mind when we talk about the algae is one is that's why a lot of the seaweed inputs work so well, is that
Well, that's a good thought. That's a good idea. Yes, the seaweed extracts have bacteria in them. They are typically not pure seaweed extracts. They usually have bacteria floating around inside them. And some of those bacteria in the seaweed extracts are very likely from endophytes in the algae, right? So these algae, these brown algae, and typically it's brown algae that are used for seaweed extracts. So those likely have their own endophytes in them, probably fixing nitrogen. I mean, we don't know the details here. This is something, I guess I'm speculating a bit much now in saying that they have endophytes in them. They use them for nitrogen, but they likely do. They likely do. We haven't had a chance to study it closely because we can't study, it's hard to study everything, but very likely they're carrying endophytes inside their cells also. They have the same needs for nutrients as other plants, even though they're growing in the water.
Yeah. So I get what I meant by that question is because we've learned that plants are also eating the algae, that I'm curious if that helps us understand or gives a different understanding or broader understanding of why the seaweed and algae inputs have been so helpful in crops.
Well, part of it is that the bacteria are in there with the algae. Part of it is because the chemistry that makes up the seaweed extract contains substances in it like chlorophylls, likely other compounds that when absorbed work as a biostimulant to trigger the plant to start being more active and getting nutrients out of bacteria, to attract bacteria. This is what we found really is that that the seaweed extracts and many of the other biostimulants will actually trigger the rhizophagy cycle in roots. They'll trigger roots to start taking in more soil microbes. And we think the reason for that is that the seaweed extracts, humic substances, biostimulants like humic substances and seaweed extracts are detected by the plant as breakdown products of microbes, of algae and plant material as breakdown products of plant material from microbe degradation of that plant material, right? So you can imagine like chlorophylls coming out or humic substances. The humic substances are actually degradation products of plant material. So plants have the capacity to detect when those substances are in the environment. They can detect it in the soil because it'll diffuse over to the soil, or you put seaweed extracts there, and then that triggers the plant. It's a signal to plants to start taking in these microbes that were degrading down that plant material, breaking down the plant material, right? So it's a, those are like signal molecules. So we think that many of our biostimulants that we use are in fact signal molecules, things that plants use to that trigger actually more rhizophagy cycle, more absorption of microbes from the soil.
Fascinating. So what I'm hearing is the biodiversity is really important. It's not just about the microbes and bacteria in the soil, or just about putting more algae in the soil. It's about having a broad spectrum of biodiversity. Would that be true?
It's absolutely correct. In fact, we did one study with Ward Labs in Nebraska, looking at Ray Ward and Willie Praetorius and a number of other people there, looking at, and also a collaboration with Walter Goldstein in Wisconsin, who's breeding this nitrogen fixing corn, right? So we did a study with Ward Labs where they looked at the fatty acid analysis in the leaves in our nitrogen fixing corn compared to some commercial cultivars. And that study showed that this fatty acid analysis, right, showed that that the nitrogen fixing plants are very high in lots of microbes that you would have in the soil, but things like gram-negative bacteria, which are nitrogen fixers, but also rhizobia, which are bacteria that are found in nodules of legumes, right? They're the nitrogen fixers in legume root nodules where nitrogen is fixed. So those bacteria actually are absorbed into the plant. Also, protozoans were found inside these leaves, the evidence of protozoans. So what that suggests is that, and I told you that algae are also absorbed, that very likely plants are absorbing more microbes than we know, as indicated by this, by our experiments where we see algae going in, and also this work where we're detecting protozoans that are absorbed into plants. So really, when you think about it, plants are absorbing many more microbes from healthy soils than we know about. And in fact, these highly microbial plants tend to be more similar to the soils and the soil diversity when you compare it to commercial cultivars. So there's a big difference in terms of the microbial content of crops that are grown on very healthy soils and that are grown to preserve those microbes in the plants.
Wow. So what does that mean for soil fertility then when we're talking about, because you said something really powerful, which was that you're noticing the microbiology of the plants that are grown in healthy soil are mimicking that soil that they're grown in, as opposed to plants that are grown in maybe less healthy soil, food web soil. And so what does that mean for soil fertility?
Well, I would say soil fertility in this case is consistent with soil microbiology, right? Soil microbiology. If you have a rich microbial soil, that means you have lots of organic material in the soil, you've got a good diversity of microbes in the soil, you've probably got some cover crops going there, or at least you try to keep plants on that soil to keep those microbes alive. And so soil fertility is really equal to soil microbial diversity with the right microbes, right? With the right microbes. It really is, I would say, soil, we want healthy soils because they're the home for the microbes, and they bring the nutrients into the plant. But once the microbes get in the plant, some of them start to fix nitrogen inside the plants, all through the plant, right? All over the plant. The plant puts them all over and then uses the microbes to trigger development of the plant. In other words, if we took all the microbes out, the plants can't grow at all. They can't, they grow very little. They'll die, they'll succumb to disease, and we can take them out by just sterilizing seeds, and we can get 80% of the microbes or 70% of the microbes off by sterilizing seeds. And you can do that just with Clorox, sterilize them a long time, clean that, and then grow that, and then you have a plant with very few microbes on it. Grow it in a soil with few microbes in it, a sterile soil, we can do that. And when we do that, plants show almost no root development, very suppressed roots. They will show low shoot growth, and they will show high susceptibility to disease. And they basically will die after a little while, especially if we do the same treatment. And then we put, with one of them, we put microbes back on, right? Just put one species of microbe back on that came out of it, right? We put it back on, and the difference is like day and night. You have full healthy roots compared to little tiny suppressed roots, shoots that are small, disease that happens, like fungi like fusarium will come and they'll take out the non-microbial plants, but the microbial plants are not affected. So it really makes a big difference for the growth of the plant. And the fertility, I should say, especially where it comes to nitrogen fertility, really happens inside the plant and not in the soil. It's actually a phenomenon of the plant. This is how it naturally happens, right? There's a little bit of nitrogen that gets absorbed from the soil, and we know that can happen. We know that's how our fertilizers work. That's what we put it on, and we can get it in. We know we can do that. But actually, what plants are doing is they're bringing the bacteria into the cells and into their tissues. Those bacteria are producing growth regulating substances, things like ethylene and nitrogen. And those act as biostimulants and growth hormones, and they cause the plant to grow. So the growth that you get in terms of larger cells, in terms of larger roots, in terms of larger everything, is largely because of the growth promotion that's happening in plants, because those bacteria are there providing those substances. And that's really how plants grow. There's no plant that grows free of microbes. There's no plant. This is how plants grow. This is what they do to get their nutrients. This is how they're healthy, as they absorb these microbes, and then they nurture them, interact with them inside their cells. That's how they work. So it's really what it means is that's the way plants grow. They don't grow by putting fertilizer. That's very unnatural fertilizer, chemical fertilizers. We can grow them like that, but that's not the way they work. That's not the way they become healthful and full of helpful nutrients. The only way to do that, for them to become healthful and full of nutrients, is to use microbes to grow. And that means we have to limit. It doesn't mean we have to totally remove our other fertilizers, but we have to drastically curtail them, because what happens when we try to grow plants with fertilizer, they'll still have some microbes, but we shut down the process and they stop absorbing new microbes. And it really affects the whole plant. I mean, it affects, there's another effect that I didn't talk about, and that is that when microbes are in a plant, they become much more variable. They may grow a little slower. They're not like growing like a bat out of hell. They don't grow as fast as when we squirt liquid fertilizer on them, but they grow well, they stay green, they grow a little bit slower, but they produce more nutrients. They absorb more nutrients, they produce more nutrients internal. And I mean, the microbes are producing nutrients, but the plant is also producing nutrients because those microbes are there, because there's an interaction with the microbes inside the plant cells with a reactive oxygen. The plant cell produces reactive oxygen, puts it on the microbe secretes nitrogen out to try to counteract that. But this interaction with the plant cell using reactive oxygen causes the plant to upregulate, to increase its content of antioxidants. And those antioxidants are all those helpful compounds that we like to eat and put in our bodies, right? The phenolics, like we get out of chocolate and we get out of other kinds of foods that are rich in phenolics. So the healthy stuff, that's what the plants make because they have microbes, and they don't make that. They do not need to make that, and they don't make it if we grow them with fertilizer. They also are not as hardy. They're more susceptible to disease if we grow them with fertilizer. If we grow them with microbes, they're healthier. They're more resistant. The microbes can also protect them from that disease. So,
Man, I'm getting so many visuals as you're talking. One was how powerful, just how powerful plants are, first of all, to be able to take one piece of their nutrition puzzle and turn it into this web of nutrition that makes them stronger and better. So that's like building your immune system, right? It sounds like the bacteria is connected to the plant's immunity. And so often we're used to going externally, right? Externally to treat our plants, to treat our soil, to get rid of the disease, to get rid of the pests. And what we've been doing is actually making our plants weaker. So we're taking away their agency. We're taking away their power to be strong by throwing all this stuff at them, when in fact, if we give them the foundation that they need, which is the microbes and the all the good biology, then they can actually consume that, they eat that, they bring it up into their system, and they make it multiply. So it's the visual that came into my mind is like when you drop a drop of food coloring into water and just goes everywhere. That's almost like what that bacteria does for that plant. It just
it becomes a
goes
part of
everywhere.
becomes a
Yeah.
part of the plant, becomes a
Mhm.
part of the plant. It's more than that, though, because what it is the plant cultivates these bacteria inside their tissues, inside their cells. So the plants are cultivating these microbes. It's not, it just gets in. The plant is actively cultivating them. A few microbes will be taken in at the root tip, into those cells at the root tip, and then the plant cultivates those microbes internally, makes many, many copies of them, and then shoots them out of the root hairs back into the soil. At growth spurt, ejection of microbes, a growth spurt, an ejection, a growth spurt, an ejection, a growth spurt, an ejection. Thousands of bacteria go out of a root hair, whereas dozens may have been absorbed in, right? So the plant gets the nutrients out of those microbes that come from the soil, then it takes the microbes that are remaining inside those cells, inside those root hairs, and then it starts processing them and extracting nitrogen from them. And we can see the nitrogen coming out with these histochemical stains that we use in these root hairs. So the nitrogen actually comes out of the bacteria inside the hairs, not outside the hairs. We think of root hairs typically as these things that go out to just absorb nitrogen and fix it in the soil. That's not, I mean, they might do a little bit of that, but but actually they're getting nitrogen from the bacteria inside the root hair that they're cultivating in the root hair. So the same thing, that same thing is happening in the leaves, in the epidermal cells, in the leaf hairs, and even and this is gonna, I don't know if this will blow your mind or not, Natalie, but it blows my mind. These bacteria will actually, in like this corn that I was talking about, this nitrogen fixing corn, they actually go through the pollen grains. So the bacteria, we can see them inside the pollen grains, and we can see them actually in the pollen mother cells. And so the pollen mother cells are the cells that divided to give rise to the pollen, right? So there's what we call meiosis happening inside these pollen mother cells to result in the pollen grains, right? And the cell divisions that are happening in there are important for variation that happens in the corn plants, the seed, right? Because this pollen grain, you get variation, and the because the bacteria are there, the bacteria may be causing increased genetic variation in this corn plant, because we can see the bacteria in the pollen mother cells. We can also see nitrogen, nitrate, and ethylene produced in those pollen mother cells. And this goes into some literature, other literature, that some other work that some others have done. They've looked at ethylene and nitrate, and those, both ethylene and nitrate, will trigger plant nuclei to divide in a process, DNA replication of the plant, right? In a process that's called endoreduplication, or simply endonuclear replication, like that. Basically, all it means is that the DNA, the chromosomes, divide. But that's important for the plant genetics, because if the endophyte is causing DNA replication in these pollen grains and the pollen mother cells, it means the endophyte is causing more variability, more variation. And plants like corn, they have these called transposons or jumping genes. You've probably heard about that, but they're just elements, DNA elements that move around, right, and cause genetic differences in genetic expression, genetic changes in the plant, right? So plants with more endophytes are going to have a higher capacity to show variation and to evolve, to select, to adapt. If you have a rapidly changing climate, endophytes are going to impact that in a positive way. They're going to increase the ability of plants to adapt, and crops to adapt this highly changing, climate changing situation. Plants are going to, so endophytes are critical. We think now we have to go in, we have to go in, and what we have to show next is we have to show that, in fact, in our plants where we see endophytes, we not only see the chemicals, but we also then show that those plants are showing more DNA replication in those cells where we're having the stimulation by these microbes. So, but that's what it looks like so far. And we can see these microbes going actually into the nuclei of the plant. And they go into the nuclear envelope. So this effect on plant variability, plant evolution, may be a real thing, and it may be very, very important. When you think about, Natalie, when you think about our crops, right? You think about our corn, for example, our commercial cultivation systems, where we have big, massive amounts, and we're using lots of fertilizers, and we're using agrochemicals and stuff, and we're not using microbes. Those plants are perfectly manicured, right? They're perfectly manicured. They're the same size, they're the same amount, they die at the same time, if if you have a disease that comes out, it'll knock out most of those plants. You'll get them spreading. But if you have a biological rich plant, say that you're growing on healthy, microbial rich, healthy soils, microbial rich soils, uh, and especially if you've grown that plant in that soil for a couple of years so that you can acquire the good microbes, right? Then you're going to see plants, the offspring of plants, becoming more variable. You're going to see more variation, more variation. They're gonna have this wild look, the wild look. These land race corn that people use as land race cultivars are not these, you know, identical plants. They're variable. They're changing. They're changeable. They'll have leaves will stay green longer. There'll be bigger leaves. There'll be bigger trichomes. There'll be bigger root systems, more branching root systems. They'll be, if you look at them with a microscope, they'll be full of microbes in the tissues. It's not hard to show that. Not hard to see them. All you got to do is look, microscope.
That does blow my mind. And what even blows it more is just the intelligence of plants and nature is so awe-inspiring. It really, I mean, you must just at times pause when you.
Well, I do. I pause, and the reason that I keep doing interviews about this is not because I like to hear myself on YouTube or wherever else it is, right? It's because I think this is information that's really important for all of those people that are trying to do regenerative and organic and biological agriculture, and they didn't really understand what's happening, right? They did it, they went about it to do this on faith, or based on what evidence they have, in spite of the fact that most of the published science and most of the people that are advising them are telling them, use chemical fertilizer and do it this way, and they're trying to buck the system, right? Well, what I have to say, my message to all of them is, you were correct. You were right. Plants have to have these microbes. They're microbial cultivators. They get their nutrients from microbes. They get their health from microbes. Their immune system is directly from those microbes. Their nutrients are from those microbes. So, and I think that, and I consider that very important, supporting that group, because it's that group, the regenerative agricultures and the organic agricultures, that are going to actually change agriculture. So what support that I can give to them in this biological agriculture community, it can help, I think, them to be successful in changing agriculture, and for the sake of human health and the sake of environment and the world, and better everything in general. Yeah.
Gratitude. Thank you. Thank
Yeah.
you. Thank you. Thank
Yeah.
you. Thank you. Yeah, my dad is just so grateful for your voice and the research that you're doing. And I mean, there's many other people in this space that he's just so grateful for, because he sees the value of you showing and demonstrating what people have been practicing in biodynamics and organic, and even the Korean natural farming and different practices have been fostering a different way of doing things, of working with nature instead of trying to control nature. And so, yeah, there's a lot of gratitude for you. So thank you for creating space, for sharing what you're learning, and also for just keeping on with what you're doing, because it's important.
Well, thank you so much, Natalie. And it really is a lot of people doing this, including you, all of us, working in this. But I'm so glad that the work that we're doing at Rutgers can support this movement. And hopefully, hopefully, give some inspiration, right? Give some inspiration and some confidence that with regenerative agriculture, we're on the right path.
When you say regenerative agriculture, what does that mean to you?
Biological agriculture,
Okay.
organic agriculture,
All right.
fixing soil. It doesn't, I'm not very sophisticated.
I was just curious.
I'm
I
not
was
very
just curious.
sophisticated. I'm not
Yeah.
very sophisticated. I actually, I was fascinated to learn about Korean natural farming too, and some of the practices and how rhizophagy cycle does explain some of those practices, moving microbes, moving endophytes, into plants, and the compost teas, and how it all works. It all makes sense, if you look at it from the what it's doing to rhizophagy cycle and stimulating plant growth through rhizophagy cycle. It all makes sense then.
It really does.
Yeah.
And what excites you the most about what you're learning right now and discovering?
It's radically different. It's radically different. We didn't think that that we would see this in plants, not to this extent. I mean, I've worked on endophytes for 45 plus years, right? A long time. Okay, but to see the extent to which plants actually absorb microbes and then cultivate those microbes internally in their cells, to me, it's mind-blowing. It's absolutely mind-blowing. The fact that we haven't found it before, it took 2010 before someone saw these microbes going into plant cells. That was the Australian group that I talk about in Queensland, Australia, who named rhizophagy, right? They named that. And they saw it first. It took in 2010 before people could see it. And I think it was because people don't have a concept for it, right? There's no idea. In fact, if you see a microbe in a plant cell, we assume, well, microbes are bad. It must be a pathogen, or it must be a contaminant, or something like that. You actually never think it's a microbe inside that plant cell. And the other thing that's apparent, it's not just that they absorb them, but they process them, they interact with them, they process them, they use them for development. And then that goes even further now, because we have a group now at Rutgers with another professor and some other students that are looking at these kind of associations in the very earliest plants on Earth, the simplest plants, the mosses and the liverworts. The first plants on planet Earth had these microbes inside their cells. So this is not something that just happens with some plants. This is something that's been happening with all plants from the very beginning of plant existence on Earth. It's something that's been from the simplest plants to becoming only more and more complicated, and the more complicated seed plants, the more developed seed plants, the more evolved, they do it too, but in a more sophisticated manner, in that they have trichomes, special kinds of trichomes, glandular trichomes with chemistry in it. They have root hairs that cycle microbes out of them, whereas some of these more primitive plants don't make root hairs. They just absorb microbes and juice them then for nitrogen, interact with them to get nitrogen out of them. Okay, but all plants have been internalizing microbes from the very beginning. And this is something that humanity just missed. All of us missed it. All of us missed it. All of us missed it. We moved past it. We moved to physiology. We moved to molecular biology. We moved to genomics, metagenomics. We passed up this basic biological interaction between these microbes, these endophytic microbes, and the plant. And so all of that we have to now look at in a very detailed way to understand how it all works and how it, we have just the outlines now of how things work, but a lot, you know, it,
Mm.
I wish we could get more and more people in doing this work. It's not like we're trying to um, monopolize work in this area. We're not. We're not. There's so much to do that it's going to take more and more people looking at these kind of associations inside plants to really understand how to manipulate them. We're just touching the iceberg now, but I mean, I think this is going to be in the future, this is going to be a big way in which we do agriculture, I think. At least it looks like an opening for us, right? Looks like an
Right,
opening for us. Looks like an
right.
opening.
And is that kind of connected more to the terrain theory as well?
Tell me about the terrain theory now.
Well, what I mean by that is like, you said something really curious and powerful, which is how we jumped from um, what we understood about plants to the inputs and the chemical connections of plants and treating them from the inside out. I'll say treating them from the outside in. And so now we're looking at the intelligence of plants from the inside out. And so that to me is more connected to the terrain, instead of like the germ theory of like, let's treat everything to make everything safe and let's give the plants what they need so they can grow strong, and let's kill what's in the soil so that it's not a threat to the plants, to what I'm connecting to the terrain, which is let's looking at the intelligence of nature, the intelligence of plants and their natural terrain within and around them to be robust, to grow and to absorb and to evolve, so to speak, as you're saying about the pollen on the corn. That's what I
I
mean.
think I think it is too. I think that yes, I think sometimes you use big terms that I don't quite follow.
Mm.
And so your terrain was one that I didn't, that uses of that term I didn't quite follow. But yes, what is incorrect, or at least it is something I can't say. um, Microbe theory of disease is incorrect. We know it's not fully correct. We know that
Mhm.
already. But all microbes, and we know for certain, all microbes don't cause disease. And in fact, most microbes don't cause disease, and plants and animals benefit from those microbes. In fact, they benefit. They benefit humans and plants and fungi. Mushrooms too absorb bacteria into their hyphae, just like plants do. They don't have
Mhm.
rhizophagy cycle, but they still. So all of these higher eukaryotes absorb these microbes into their cells, and they deal with them in different ways, but they absorb them in, and they're beneficial, right? So the terrain idea, meaning microbes in the environment, that plants are using all those microbes in the environment, and even the ones that cause disease, if there's enough microbes there, they don't cause disease. Things like fungi that are pathogenic. When the bacteria are there, the fungal behavior changes. Things like fusarium, for example, that would normally attack a plant. But when there's lots of microbes in that plant and lots of microbes in the soil, the microbes go inside the fungus, inside the fusarium disease fungus, right, inside the hyphae of the fungus, and then change the behavior of the fungus, cause the fungus to be less virulent. That it doesn't grow, the growth rate will be reduced, and it doesn't attack the plant. It's the situation where we remove the microbes from the soil and we remove the microbes from the plant, that fusarium then attacks the plant and destroys the plant. Okay, and hence the deal on our uh, on our beautifully cultivated corn, commercial, chemical fed corn that uh, that where we've compromised the microbiology, and then now those plants are highly susceptible, and they're all highly susceptible to the disease because the microbes aren't there, and those microbes alter behavior of the fungus, they alter behavior of the pathogen. And so we see that in human gut too. We see the same thing happening.
Right. And I think one
Yeah.
of the really important pieces to take away is it starts at the seed level, right? It's starting at the seed level. It's before the seed even reaches the soil. It's how that seed has been treated or not treated, or sterilized or not sterilized.
It does.
Um, there's a couple more things I just want to touch on before we wrap up. One is, can a sterilized seed that's been stripped of all the healthy microbes and all the microbes, period, will that seed thrive in a soil that is rich in fertility and biodiversity, or does it struggle right from the get-go?
Well, there's a little bit of struggle. There's a little bit of struggle, but it doesn't last very long. As soon as the seedling, if there's no microbes or few microbes, or microbes have been lost from seeds, they'll germinate. Typically, if the seed is on the surface, right, they'll germinate. Even if they're not, if they're down in under the soil near an open space, or they may not be immediately microbes to colonize it, right? Okay, but once it starts to grow, and the roots will grow, and they start going where the soil has more microbes, then they can acquire those microbes directly and get them back. So yes, they can get them all back.
So a
Now,
sterilized seed is not all lost if that's all the person has access to.
yeah,
They
it's not all
start with that.
lost. In fact, you could get these like commercial cultivars of corn and grow them for a few years in healthy soil and recover their capacity, their growth, their microbes, recover some microbes. Now, what's happened in nature, though, with these land races, is they keep getting microbes, and they keep getting the ones that are good for them, right? They keep getting, so they go years and years. However many these indigenous farmers have been growing this corn on their soils, they've been taking these microbes, so they're getting better and better microbiology, right? So if you get rid of that microbiology, then you put it in a soil where you've got good microbes, it can grow, but they may not be as good as the ones that the plant got over 100 years where it was growing in a healthy soil, right? It had all that time to keep selecting the best ones. Yeah, so we can damage them, we can get them back, we can get them, grow them in a good, healthy, biological rich soil, grow them there for a while, 3 years, 2 years, and then they're pretty good, they'll get back. But to get those great microbes, it may take growing for years, to maybe in some different environments, to sample the good ones, right? Like that.
Right. And that's why seed saving is so powerful when we do seed saving on the land that we're growing on, right? It's because it's
That's right. That's right. And it's also terrible that there is an effort in some places, some countries like Africa, to prevent farmers from saving their seed, or giving away their seed, or giving their seed, or reusing seed. That's terrible, because it's kind of an ignorant move. It's kind of an ignorant move on legislatures and even big ag, because big ag wants to control the seeds, right? They want to control the seeds, but it's a bad move, because what happens is, with these, go back to the variability, right? These plants that are produced to be perfectly, to work with chemical fertilizers and perfectly uniform, they are not hardy, and they don't show the variability that it'll take if the big pathogen comes along, something really bad, or if we have climate change gets so bad and we just keep losing crops. We're going to need that variability that comes from plants that have endophytes in them. And so really, for developing new crops. We're really for changing environment on Earth to kind of give us some insurance to keep people from starving to death, from having these cataclysms that result in starvation, massive loss of crops, starvation. We really need to start growing plants with microbiology in healthy soils and start working to get these plants, to get these microbes back into plants. They'll be hardier. They could take the stress, the stress. They'll require fewer fertilizers. They'll be richer and healthier. They'll be healthier for people. They'll improve our microbiomes because our microbiomes actually come from foods we eat, from the endophytes in the plants that we're eating. And then that'll result not only in the nutrients, more nutrients in the plant that we're getting, all those helpful phenolics and other nutrients, and but the antioxidants and that improve our health, the phenolics and so forth. So it's really critical. It really is critical that we start to grow crops biologically. Doesn't mean everyone has to do it, but certainly we're going to have to start in a much, much bigger way.
So be it, and so it is.
Yeah.
So one other piece I heard you touch on was, and not today, but in another interview you did, was the electrical charge that's happening at the root tips. And so can you just tell me more about that? Because I'm all about energy, and I'm very curious about that, having studied energy as a energy practitioner in complementary medicine. So I'm curious about how that's showing up in what you're seeing in the rhizophagy cycle and in bacteria and plants.
Well, this is something that so few people study, to be honest. So few people study the charges on plants and the role that has. We started to look at it because we're trying to figure out how these bacteria are being absorbed into the root tips, right? And so, there's a different hypotheses, and one of those hypotheses is that they actually are being pulled in by electrostatic charges. And that is that root tips have a net negative charge on the tip. And bacteria tend to, I think it's net positive, something like that. I don't know that it matters. But the charge, we think, is one of the factors. And of course, they can swim to the roots following exudates and so forth. But we think the charges bring them in to that tip. Not totally certain of that, but it's one possibility, and it is known that there are charges on these root tips, and so it could explain that. I mean, this is this whole area of charges in cells and in organisms. People don't really understand that, and we don't really study that. I mean, you remember the little experiment where you take a potato and you put a light bulb in it and it lights the light bulb, right? Cool little phenomenon, right? That means something. That means something. It means something. There's a process going on that's reflecting, something, and we may not know what it is. We see the same thing on roots, charges on the tips of roots. That means something. There's a reason for that. That's actually doing something, So the stuff, and this is why I'm open to practices that people might be doing that we don't understand yet, right? That we don't understand, because I know that it may just be a matter of time before we do have an understanding of it, before we understand the mechanism of how something works. And I think it's important. I think that is very important. I mean, like I say again, that's why I keep giving these talks or interviews, is because I want to send the message out with regard to what's happening in plants. And I think it's important for people, for because people have agency, right? And I want to make sure that they have agency. I'm hopeful that what the work we're doing will give that agency and give the understanding of how things work, so that people can then make the changes that we need in agriculture.
Mhm. And what does biodiversity cultivating life mean to you?
Well, nature is biodiverse, right? I mean, how can you not? How can we want to destroy nature? I mean, sometimes we essentially are, right?
Mm.
We essentially are. But and what do we see? What do we see happening? Biodiversity is collapsing, right? So it directly has to do with, biodiversity has to do with nature and the health of nature. And so what we have happening now in our systems on Earth, in our bodies, is we have a collapse of the natural systems of, we're losing biodiversity, we're losing the microbes in our bodies, we're losing the function of those microbes, we're losing the function of the microbes in our crops. We're putting chemicals out to the environment that's knocking plants out. We're really destroying the systems that keep us alive. And so so this biodiversity is a direct reflection of Mother Nature, the beauty of nature, the functioning of nature. It's not just something that they're systems, not just something that we can measure and assess and, oh, we want to keep it. It's functional. It's functional. It's like a cover crop. The more cover crops you have for a crop in there, the more you have, the more diverse it is, the more diversity in the endophytes and the microbes that are going to be in the soil. And then the better that plant is going to grow, right? That biodiversity, this is the natural biodiversity that Mother Nature created. This is how plants work. This is how nature made plants to work. That's your agency. That's the other big word that you used that I enjoyed, the term, right? It's not really a big word, but to refer to plants as having agency, I love that. They do have agency. They do have agency. They do function on their own. They do things. They have their own way of getting nutrients. They have their way of making their cells grow. And it all goes back to the microbes.
Oh, I just love that. I love how you summarize that, James. It's so powerful. It's just so powerful and gives so much hope and possibility to where we are today and where we have the potential of growing into. And in closing, what's one challenge you would give or gift to people, to farmers and growers? What is a challenge you'd give them?
I want them to see the microbes in the plants, to see them, to see for themselves what actually is happening. And I think that if they see it, and so we just, me and a couple of NOFA guys, uh, uh, John Duke and Ruben Perilla from Massachusetts NOFA, right? We just did at Acres, we did a, a microscopy session where we looked at soil microbes. We showed the whole group what these soil microbes look like, what healthy soil microbes, what they all kinds look like, and what rhizophagy cycle, microbes coming out of roots, look like. And I wanted them to see it, because I think if they see it, it'll change how they think about growing crops. It'll plant a seed, right? It'll change how they think about it. It'll plant a seed. It'll make it so they start to think, well, plants, why don't I use that and see if that really is working? I see the microbes there, and it's not. And I think it's extremely important that, this is a people's movement, biological agriculture, right? All the terms that we used, right? To me, it's all biological agriculture, with different pieces
Yeah,
of clothing on it, right? But
absolutely.
it's all biological agriculture. That if people see how plants are actually doing it, if they actually can see it with their own eyes, then they'll believe it. And it's not like our extension or our science that we go to meetings and we show the data, the tables of data, and all of the stuff that is complicated. And then we know, oh, we are all humans. We are all smart, right? We're all smart. Look at all the data, look, great data. But actually, we are all smart. And we don't need the complication. We don't need the complication necessarily of all the tables of data to see what's going on. If we can just see inside the plants, see what's happening with the microbes, the soil microbes becoming plant microbes, how plants work with those microbes, how they require those microbes, then with our own eyes, when everyone sees it with their own eyes, then it'll be easier to implement. And it'll be easier to resist the pressure to use a lot of fertilizer, right? If you see with your own eyes, you see it yourself. You don't have to take my word for it. You see it. That's what I think is important. That's what I'm trying to do. That's why I like to show the slides. But even beyond that, I I want to show from the plant itself so that
Mm.
they can then put that into the microscope, and then we'll look at the microbes inside the plant so they see it themselves. You know what I'm saying?
Oh, yeah,
It's
that's cool. So James, will you share those slides with us so people can
sure.
see them
Yes.
too? Okay,
You
well,
mean now or when?
well, I just want to honor your time because we've been going.
Yeah, no, I'll send them to you. I'll send you some
Okay.
slides. You want me to send you some slides? Or do you
Yeah,
want me
yeah.
or do you
Um,
want me to share something?
do you want to share one quickly and we can share one it's not
No, because
no second.
because I'll just send it to you. And
Okay,
it sounds like that's what you're gonna do, right? Put it,
perfect. Yeah.
I'll do that. I'll send you some.
Thank
Yeah,
you.
you're
And then people
welcome.
can actually see it.
You can see them. Yes,
Well,
yes,
that's
yes.
awesome. Thank you.
Thank you, Natalie.
Thank you. Well, thank you for your time today, Dr. James White, and I thank you for your curious heart and the wonder that you bring to the conversation of plants and biodiversity and rhizophagy and all that you're discovering and the way that you share your research with us and your perspectives and also your curiosity. It really is inviting. It makes these conversations more understandable, and it makes I think it really, the way you share the information really gives us like a springboard or a place to jump off of, and to have confidence in doing what we're doing, in whether we are used to using chemicals in the way and inputs in the way that we farm. It creates an environment where we're like, okay, I think I can do this, and know that, and have a bit of faith that what I'm going to do differently is going to work. And then it also gives like you said, agency to people who have been blindly organic or biodynamic or Korean natural farming from observation. So blindly in terms of science, but eyes wide open in terms of observation, right? So you're just like this beautiful beacon of light in this space. So I thank you.
I thank you too for saying that, Natalie. Thank you so much.
You can follow Professor James White on LinkedIn, or you can find him at Rutgers University under the Department of Plant Biology. Go and check out James White's slideshow so that you can see the rhizophagy cycle yourself. And if you can, get yourself a microscope or get yourself to a microscope course so you can see all the microbes under the lens, amplified, moving around and dancing. And most of all, enjoy the process. Enjoy the process of building biodiversity in your soil, growing healthy, nutrient dense crops, and creating an environment for life to thrive. and have fun doing it. Big love. I'm Natalie Forsbauer, founder of Heart and Soil Living and host of the Soil Summit 2024. Make yourself an amazing day. We deeply appreciate the work you are doing. You are amazing.